Hi, so my name's Gareth Morris. I'm currently both a lecturer in neuroscience at the University of Manchester and a senior research fellow funded by the Epilepsy Research Institute at University College London. I studied for a PhD in epilepsy research at the University of Birmingham, working with John Jeffries, probably about 10 years ago now, I think I finished. So that was really my first foray into the epilepsy field. The microRNA aspect more began when I moved down for my first postdoc at UCL with Stephanie Shorge. And really we were studying microRNA as part of a EU-funded European consortium. And the idea really was to sort of probe all these questions about can microRNAs be used to therapeutic targets, as biomarkers, and so on in epilepsy. So that's really where I started combining the two aspects. Since then, I moved over to Dublin on a Marie Curie fellowship, working with David Henschel. And that's where I really focused quite heavily on microRNAs in epilepsy. And, we discovered a few nice ideas there around sort of therapeutic targets, particularly how we can manipulate microRNAs and tweak things like voltage-gated sodium channels, which then feeds in as a therapeutic target for epilepsy. So I suppose there's a few advantages that we can talk about. So one alternative model would be something like cell culture in vitro. And the real advantage of brain slices there is that you retain a more realistic physiological circuitry. So I work on the hippocampus mainly, and you can retain those really realistic hippocampal connections, which are quite unique and quite pretty to look at. And you just can't capture that using cells grown in a dish. The sort of other direction would be to use whole animal models in vivo. The advantage really of brain slices, I would say, I suppose there's a few points. There's technical advantages. It's easier to visualize individual cells in a brain slice. It's easier to target those cells within the circuit. It's easier to apply compounds very quickly. And you can, you get multiple brain slices from one brain, so you can apply different compounds to effectively the same brain and make comparisons there. There's also ethical advantages. Obviously, you're not using disease models in animals, so it's great to reduce the number of animals used in that way as well. So I think one of the most exciting discoveries that I've been involved in recently, and I touched on this in one of the earlier questions, is finding a microRNA that interacts with voltage-gated sodium channels that we can use therapeutically. So this came about from work that I was involved in David Henschel's lab over in Dublin. And it was the PhD project of Mona Hyland, who was an excellent student and now is a postdoc over there. And what we discovered essentially is that there's a particular microRNA called microRNA 335, which is dysregulated in epilepsy. So that sort of hints at some epileptogenic and disease mechanisms. And what we found ultimately is that if we overexpress that microRNA using a sort of viral vector gene therapy type approach, we can actually reduce the expression of voltage-gated sodium channels in the epileptic brain, and that reduces the excitability of the neurons and so reduces the sort of propensity to generate seizures. So it's just quite a nice therapeutic link between microRNAs and their molecular effects and epilepsy. Yeah, so I think one of the things that strikes me as being a sort of promising future direction, and actually is already starting to happen, is really the use of human brain slices to research epilepsy. So how that works is that some people with epilepsy undergo a resective surgery in the hospital to actually remove the part of their brain that is causing epilepsy. And with their consent and their permission, we can actually collect that piece of brain tissue for research. We can handle that effectively in the same way as a rodent brain or an animal brain. And so we can make brain slices from this real human tissue. And the advantage is then we're working with a piece of tissue that has human genetics and sort of human neurobiology and so on. And so that's just a really powerful way to actually test our therapies and make sure that they're likely to work for people, humans that have epilepsy in the clinic.